Hi, everybody. As mentioned, my name is Joanna Pineda, Master's Candidate, Simpson Lab, and I'll be talking about my research exploring a haplotype-based approach to calling somatic mutations using linked read sequencing data. So a general overview of the work that I'll be talking about. This project is stemmed in collaboration with Dr. Trevor Pugh's lab, where we're trying to understand the genetic etiology of glioblastoma. And the part of my project is trying to call somatic mutations with a tumor-only approach using a haplotype-based method and leveraging the phasing information from linked reads. So a brief overview of genetic differences. They're germline variants or somatic mutations. And germline variants are inherited from a parent and are found in every cell, whereas somatic mutations are acquired over time. And the reason why we're interested in somatic mutations is they're known to drive cancer. So somatic mutation calling is the task of trying to accurately detect these mutations specific to the tumors. And this involves distinguishing them from germline variants and artifacts. The traditional approach of doing this is using the matched tumor-normal approach. And so how this is done is from a single individual. We take a DNA sample from their tumor tissue and a normal sample or a healthy sample, which is often their blood, pass it off to a DNA sequencer, and we obtain a set of reads that we align to a reference, and then we're able to identify differences, and I've highlighted them in these different colors. The variant caller is then tasked with trying to categorize these differences as either being a somatic mutation, a genetic or a germline variant, or an artifact. The way that it's done is they usually assess the variant allele frequency, or the VAF. And so in this example here, with the orange one, for example, there's 2 reads there that are evidence reads or have evidence for the orange variant. And there's 10 reads there in total. And so the variant allele frequency is 20%. Germline variants are distinguished because we know that they're approximately 50% or 100% for heterozygous mutations or heterozygous variants or homozygous variants. The problem is, how close does it need to be to the expected in order for it to be called a germline variant? In the tumor-normal approach, we can leverage the normal sample's information. So we can see that this blue variant here has evidence in the normal sample. So that gives us more confidence that this is a germline mutation. Somatic mutations can have a varying VAF. The problem is that they can be very low with their VAFs, and that's similar to errors. Other approaches use a tumor-only methodology. So as the name implies, we don't have access to that normal sample. This is interesting because we no longer need to sequence the normal sample. This task is even more challenging for factors such as tumor purity, where sometimes the normal sample can be in the tumor sample, tumor heterogeneity, copy number variation. So here's a little GIF that I made that demonstrates the tumor purity impact. It lowers the variant allele frequency, giving us more of a challenge between distinguishing somatic and errors. More recent approaches use this haplotype-based methodology. So this differs from the mainstream methods, which is a position-based approach. And the way that they do this in the position-based approach is they assess each difference independently of the surrounding mutations, whereas the haplotype-based approach uses this phasing information. So that's using the surrounded co-inherited variants to assess these differences. Little work has actually leveraged the phasing information in tumor-only calling. And the property that we would want to use when we use a haplotype-based approach in this tumor-only calling mode is that somatic mutations segregate onto one haplotype, whereas errors do not. And so if we know the phasing information, we know which chromosome the reads are coming from. And so in this example here in the orange, we know that this segregates, we see that it segregates onto the maternal haplotype. And so we're more confident that this is a somatic mutation. And the same goes for this pink one here. This one, we can see that we have evidence for it on both haplotypes. And so this is most likely a germline homozygous variant. What's interesting is this case here, where it doesn't segregate onto haplotype. And so this is giving us, suggesting that this is an error. And the final one here is a somatic mutation because it segregates by haplotype. And so as we can see in this example, using haplotype information can be really useful when calling somatic mutations. The way that we resolve these haplotypes is we use these heterozygous sites. And so here at these two loci, we see that there's evidence for A and C at the first one, and there's evidence for G and T in the second loci. And we want to know if A is inherited with G or A is inherited with T, and the same goes for C. The problem with short reads is that we don't have any reads providing evidence to any of these haplotypes. The advantage of long reads is that we can see that they span these heterozygous variant sites, and we gain more evidence in these two haplotypes. So 10x Genomics generates linked reads, and they're a short read sequencing data type at its core. What's cool about them is they utilize molecular barcodes, which allows us to derive which DNA fragment a read comes from. And so the way that it works is we break up the genome into high molecular weight DNA fragments of about 50 to 100 kilobases. Using their microfluidics device, we're able to get these high molecular weight DNA fragments into these gel beads, which have these barcodes. We end up with barcoded DNA fragments, and we pass it off to a short read sequencer. So in the end, what we get are barcoded reads, and because we're able to derive which DNA fragment this comes from, we get that long-range information that we want in order for us to resolve that haplotype. So again, the motivation behind this project is, can we leverage haplotype information provided by linked reads to call somatic mutations in tumor-only mode? And so here's an IGV screenshot of a true somatic mutation. And so the bars here are indicating the reads. The differences are indicated again by the colored boxes. And in the center, I've highlighted that true somatic mutation. The variant allele frequency is pretty low, and without that phasing information, it's hard to tell if this is true. But when we use the phasing information from the linked reads, we can see that it segregates by haplotype. And so here we're really leveraging this idea that errors are most likely not going to be hitting one haplotype alone. And so the next question is, how does calling somatic mutations compare in linked reads and in regular reads? And so we obtained some data from a patient with glioblastoma. So we have a primary sample and a recurrent sample. And so we have linked reads for that. And so then we can pass it on to a somatic calling pipeline. We could do a tumor-only mode calling for the primary and the recurrent. For that same sample, we also have Illumina reads or regular whole genome sequencing. We can also do that tumor-only calling for these two samples. We also obtained a normal sample, which enables tumor-normal calling. So we analyzed the number of candidate somatic mutations called in these different modes with these different data types. So we see that in the tumor-only mode, for the linked read whole genome sequencing and the regular, it's about 3.8 million. With the linked read whole genome sequencing tumor-only mode having about 35,000 more candidate somatic mutations. Mutex1 isn't actually built for tumor-only calling, so we do expect this high false positives, and a lot of this is probably germline variants. When we do look at what it's built for, which is matched tumor-normal calling, we see still this significant difference of about 100,000 variants being called in the linked read whole genome sequencing tumor. And so this gold box here is our gold standard, essentially. We expect that the number of calls would be around the regular, normal, regular tumor calls. So this prompted deeper investigation, and we applied some different filtering methods. So we used a panel of normal, we did some marking the duplicates, and we got the number down by an order of magnitude, but still it's incomparable to the gold standard here, which is 4.8 thousand. Our recent group actually built a machine learning model that does exactly what we want, linked reads, tumor-only calling. And we got about 17,000 calls from that, but it's still incomparable to what we want, which is 4.8. Okay. So this prompted a deeper investigation of these differences because we're still trying to understand why there's so many more false positives in the linked reads. We did a read position analysis. So the question that we're trying to answer here is, where do these false positives lie in the reads? When we segregated it by read 1 versus read 2, we see that in read 1, the false positives seem to be almost uniformly distributed across the reads that are carrying them. But when we look at read 2 here, we see a strong bias towards the ends of the reads. So there's obviously something suspicious here, and so we looked even deeper. When we looked at the IGV screenshots, we saw a lot of these multinucleotide variants that's consecutive base substitutions of 2 or more. In these different panels, I'm indicating the different tumor samples and the different data types. So in the top one, the top 2 here, we're seeing these multinucleotide variants only in the linked reads. We also noticed that a lot of these reads that are carrying these multinucleotide variants had a lot of soft-clipped bases. And so that's where the mapper forces the alignment there. And these reads seem to have mismatches at the very end. And so they trim these mismatched ends, and these are the soft-clipped bases. To understand where the soft-clipped bases are coming from, we ran a BLAT analysis. And so this is an evidence read where the false positive is indicated with the colors here, G and T at the end. And the slashes here indicating the soft-clipped bases. We noticed that when we ran the BLAT analysis, that the section that carries the soft-clipped bases seemed to be mapping to a nearby area, but on the reverse or on the opposite strand. To understand the underlying mechanism of these reads, we tried to do a hairpin analysis and found that a lot of the reads that are supporting the multinucleotide variants could form this hairpin structure. What was even more interesting was when we annotated where the false positive variants lie, they seem to be falling at the apex of the hairpins. So linked read whole genome sequencing uses multiple displacement amplification. Previous literature has supported that MDA can result in inverted chimeras, which shows similar traits to the reads carrying the false positive multinucleotide variants. So the next question that we wanted to answer was, how can we identify these chimeras found in linked read whole genome sequencing? So we developed a novel method called 10x trim, which tries to identify these chimeric reads using overlap detection, and the code is available on GitHub. So the idea underlying 10x trim is we noticed that a lot of hairpin structures had sequences that overlapped strongly with its reverse complement. And so we would see if the overlaps were significant based off of a score and modify the alignments to soft clip the problematic sequences. And so 10x trim would take as input a BAM file and then output a modified BAM file with the modified alignments. So for every read, we would look at the sequence, take the reverse complement, and try to detect if there's a strong overlap. And so we can give each of these overlaps a score, and this is a function of the number of matches and mismatches and gaps that appear between its sequence and the reverse complement. And this overlapping section seems to be carrying all the false positive variants. We redid the read position analysis, trying to understand where the removed variants were after doing 10x trim. And like I previously said, there seemed to be a strong bias for the false positives to fall at the ends of read 2. After 10x trim, we noticed a significant reduction in that bias. We also did a reanalysis of the multinucleotide variants. So we were seeing that a lot of them appeared just in the linked reads, but after 10x trim, we see that a lot of the evidence reads are removed or trimmed. To get an idea of how many reads were affected, we calculated the percentage of reads that were trimmed, at least one base pair. And in the linked read whole genome sequencing, there's about 8% of the reads are being affected, whereas in the regular samples, it's only about 2.5%. So many more reads were affected in the linked reads. I'm hinting again that this is very 10x specific. And this is probably the background rates, so inverted duplications or low complexity sequences that would naturally have this overlap. We revisited the number of candidate somatic mutation calls. So after 10x trim, we got about 10,000 variants removed in the tumor-only mode. We also wanted to see if this applied to other linked read whole genome sequencing data. So we downloaded some linked reads from prostate cancer samples from this paper, Viswanathan et al. And we looked at the percentage of reads that were affected in these samples again. And we see a similar trend of about 8% of the reads being affected. So a few conclusions. So we compared the number of somatic mutations being called in linked reads and in regular whole genome sequencing and found that there was a much higher number of somatic mutations being called in linked reads, prompted an investigation on these false positives, and we found a bias of false positives in the read position analysis for read twos. Multinucleotide variants seem to be linked read specific. Many of the multinucleotide variants were supported by soft-clipped evidence reads. We found some evidence of chimeric reads and potentially a secondary structure involvement. We developed a method for identifying these chimeras called 10x trim. Some future directions, we are hoping to do a minimum allele frequency cutoff. When we looked at the allele frequency distributions, we find that a lot of the false positives have an allele frequency much lower. So if we cut off any variants that have a certain allele frequency, then we can probably reduce the number of false positives further. Other future directions is maybe we can apply this haplotype sorting method to other long read data such as PacBio or Oxford Nanopore. And I'd really like to thank the members of the Simpson Lab, the Pew Lab, Taylor Lab, Dirk's Lab, ORCR Genomics, and Bioinformatics, Princess Margaret Genomics Center for all their support in my research. Thank you.